In this short tutorial, we'll focus on vaccination. We'll just look at the different kinds of vaccines and just the general purpose of them. So essentially, the whole point of administering a vaccine is to stimulate active immunity. So you artificially stimulate the immune system to produce long-lasting immunity to a particular antigen. So it's about acquiring active immunity through an artificial introduction of the antigen into the body. So the major different kinds of vaccines are as follows. So you could have a vaccine that's called an attenuated vaccine, and attenuated vaccines involve injecting live organisms, such as viruses or bacteria, into the host organism, but these pathogens have been weakened in some way by heat treatment or chemicals so that they are not infectious. And these vaccines are generally very effective and give long-lasting immunity. The drawback, though, is that there is a very, very small chance that these weakened pathogens may revert to their active infectious form and therefore cause the disease. Second kind of vaccine are inactivated vaccines, where the pathogen has been killed and so poses no threat and still has the antigens on its surface, so that will stimulate the immune response. The drawback for this is that the effect is not as great, and often it may require multiple vaccinations to build up the number of antibodies and memory cells. Another kind of vaccine is called the subunit vaccine, where just parts of a pathogen, which could be multiple different kinds of antigens that are found on its surface, so it's just the antigens that are injected into the host cell, and these of course then will stimulate the immune response. Another kind is called a conjugate vaccine, and in this case toxoid antigens may be set up so that they coat various polysaccharide molecules that would normally be found on the outside of organisms such as bacteria, and this makes those polysaccharide molecules, which are often invisible to the immune system, particularly in young infants, these toxoids actually make the polysaccharides visible to the young infant's immune system, and therefore they can develop an active immune response to them. So this involves toxoid molecules being added to bacterial polysaccharides to make these visible to the immune system, particularly in young infants. Another kind of vaccine, very recent and cutting edge, the DNA vaccines or recombinant vaccines. And recombinant vaccines. So this involves injecting DNA or getting DNA directly into a host cell, and it's the host cells that actually use the pathogen DNA to build the foreign antigens themselves, which then stimulates the body's immune system. And on the left you'll see the cycle of infection, or the cycle of what happens during different vaccinations. So as we saw in class, you would note that upon the first vaccination or the infection, the body will slowly build up a set of antibodies and memory cells, but after the infection is gone, these will slowly decline. So upon a second infection or a second vaccination, we'll see a much faster, a more rapid and greater response to the antigen, and this will produce a longer lasting effect. And in some cases you may need even a third exposure or third vaccination to build up sufficient antibodies to last you through a lifetime.